So a very good morning. What I would like to do today is to take you to the wondrous world of allometry and allocation. And just to relate to the remark that Alex Books made yesterday about the drawings, he was complaining. But in the end, my takeaway to you is that the hidden half of roots is generally comprising only 20 to 30% of the biomass. So maybe the artist didn't do that bad when he was drawing these sunflowers and these other plants. Okay. So before I would go and discuss with you the biological things, I would like to first sharpen our tools on the allocation, because during the last 100 years that has become a little bit a blunt tool. And why is that? That is because there is a lot of confusion. And so to talk you through, there's already confusion about the terminology. So let's start with photosynthetes that are produced, and sugars, when they are produced in the leaves, are either allocated to new leaves, or to the stem, or to the roots. And they do their things, and we call that allocation or translocation. And then with that, there is maintenance respiration, or there's respiration for uptake of nutrients. And so part of the sugars disappear, and the rest is used to increase the biomass of the leaves, the stems, and the roots. So people call that growth allocation or partitioning. And then in the end, people come along, chop off these plants, and they say, okay, there was so much leaf, there was so much stem, and so much root, and that's called the biomass allocation or partitioning or distribution. And why are there so many terms? That is because people try to discriminate between these three different processes. And obviously something went wrong, because some people call this partitioning and other people determine this as partitioning. So I'm going to try to make life easier for you. And like Napoleon, I think that one name or one word for somebody is not enough. You need at least two names, a first and a last name. So I will call this all allocation, but in the first case it's sugar allocation to leaf, stems and roots. It's growth allocation when you look at the increases in biomass. And it's the biomass allocation if you look at the proportion of leaves, stems, and roots. Right? That makes life, I think, a little bit easier. Now, then there's another point. Our predecessors, 100 years ago, when they were talking about allocation, they started to talk about shoot-to-root or root-to-shoot allocation. And I'm not that happy with it. And one reason is that if you have more shoot than root, then your shoot-to-root ratio can go up from 1 to 1,000 or more if you want. But if you have more root than shoots, the shoot-to-root ratio can only vary between 0 and 1. So it's a little bit of a strange distribution, and then people try to compensate for that. If they have a shoot-to-root ratio which is below 1, they talk about the root-to-shoot ratio. And then you completely get lost when some people say the shoot-to-root ratio was increasing or the root-to-shoot ratio was decreasing. So I think that's not a good idea to use shoot and roots. And there are more reasons for that. Because leaves and stems have very different functions for plants. So why would you pull that into one shoot, and why would you not take that apart? Moreover, if you use fractions, what I will do in my talk, so the leaf mass fraction, the stem mass fraction, and root mass fraction, if you're interested, for example, in fine roots, you can introduce a fine root mass fraction without any problem. Whereas if you have a ratio between two entities, you have to look about leaf relative to stem or stem relative to leaf. That becomes all very complicated. And last but not least, if I have not convinced you yet, if you're going to look at the equations that are used to describe the growth rate of the plants, the relative growth rate, it's never shoot-to-root ratio that comes up. It's always the leaf mass fraction or the root mass fraction that is one of the important components of the growth rate. Right? So I will use leaf, stem, and root mass fraction in the rest of my talk. Now, if you're going to look at the poor plant after it has been germinated, then there are all kinds of environmental variables that act upon that plant. And that's above ground, it's also below ground, and the plant has to deal with these different things and try to optimize its behavior given the different values of all these environmental factors. And a very obvious question, of course, is what is actually the effect of the abiotic environment on the allocation of biomass? Now, people are aware for a long time already that if you give less nutrients to the plants, then the plant will make more roots. And the same is when you give less water to the plants will allocate more to the roots. Whereas if you give a low light level to the plants, then there will be less biomass allocated to the roots and more to the leaves, right? That is very clear. And this person here, Brouwer, did a very interesting experiment in the 60s, because he grew various plant species in hydroponics, and he looked at, in this case, the shoot mass against the root mass. And in this allometric plot, if you halve it several times and you follow the progression of shoot and root mass, you see that you will find a straight line, which is indicated by the black line in the graph. Now, what he did is after 3 weeks, at this moment of time, where the circle, a red circle is, he removed either half of the leaves or half of the roots. And he followed in time what happens to those plants compared to the control plants. And what you can see is that within, well, almost 1 week, or otherwise 2 weeks, you see that the plants that were missing half of the roots or half of the shoots had grown in such a way that they were again on exactly this black line. And what Brouwer concluded was that there is a kind of equilibrium in the plants that makes that there is a certain amount of shoots and roots. And if you perturb it, it goes back. So it's really focused on a specific amount. And if you then give less nutrients or less light, then the plants can adjust this equilibrium. So he says there is a kind of, there is a kind of functional equilibrium, and other people that have worked on it, other bright minds have called it optimal partitioning or the balanced growth hypothesis, which say that the plant is investing to the part of the organ that is limiting growth the most. Okay, well, that is clear, but there are a number of things that are not clear. And one of the things is if we think in terms of leaves and stems and roots, how is the functional balance working if you have additional carbon? Should the plant put that into leaves, or should it put that in support and transport in the stem, or should it transport that to the roots? And the other thing is, it's all clear for nutrients and light, for example, but what happens if you give more CO2 to the plants, right? If you give more CO2, there's more photosynthase. So from that perspective, there could be more biomass allocated to the roots. But at the same time, the CO2, if the CO2 concentration is higher, the stomata close, and then you need less roots. So what will that theory tell us about an example like that of CO2? And finally, what we still don't know is how important is this change in allocation relative to the other types of acclimation that the plant can make towards lower nutrients or lower water, for example. So that's what I would like to discuss with you. And I like to give you a kind of general picture. So I love meta-analysis. And meta-analysis is cool because it gives you a kind of generalized idea across many different experiments what is happening. And you could then say, okay, is it maybe the case that some specific groups of species behave different from other groups, even if you don't have done this large experiment where you compared 60 species at the same time. So that's nice, but there is one drawback of meta-analysis I realized after doing it for 20 years. And that was that probably when we look at abiotic environmental stress, it is not the best way to analyze the responses. And I'll give you an example here. So suppose that we have 2 experiments, and one experiment done at the experiment was done at low lights compared to high lights, and we grew the plants and we measured it to certain traits. And in one experiment, the orange one, you see that this trait didn't change between low light and high light. And in a meta-analysis, you normally do a response ratio where you say, what is the difference, what's the ratio between the value at high light and at low light levels? And in this case, that would be 1, right? But in the green experiments, you see that there is a tremendous response of the trait to the growth light conditions, and the response ratio is 2. The value doubles between low lights and high lights. So in a meta-analysis, you would say, okay, on average now the response ratio is 1.5. But it could also be that actually these 2 plants or these 2 experiments were not different at all. And if you think of that, or if you look about the actual light conditions in which the plants were grown, suppose that the green experiment was about Arabidopsis, and Arabidopsis is always grown at very low light levels, right? So the experimenter thought, okay, 150 and 300, that's already quite a lot. And maybe this was an experiment with an agronomic species, and these people like to grow the plants at much higher light intensities. So in the end, it could be just one dose-response curve that is describing the response of these two different experiments. And if you're going to think about it, those response curves are, of course, superior above anything like a response ratio, because it's very generalized, and you can use it in models, for example, or you can calculate whatever ratio you want for whatever different light intensities you have. So I think it's much more instructive, and I'm trying to show you some of these dose response curves for biomass allocation. So what I've been doing the last 20 years is basically constructing these dose response curves, and I'll show you them for leaf, stem, and root mass fraction, but I do that for 100 other ecophysiological relevant traits, and I try to do that for 12 different abiotic environmental factors, right? And in each case, I look at controlled experiments where people really manipulate at only one level, so only the level of light or only the level of CO2, and where the plants have enough time to really acclimate to the conditions. And at the moment, the database has about 2,000 experiments on almost 1,800 plants. And well, there's a lot of data in it. So it is interesting to interrogate this database and see what leaf mass fraction is doing. Okay, I don't have the time to completely tell you everything about how I do the different steps, but basically I take each individual experiment. I look what is the value, in case of light, for example, what is the value of the phenotypic trait, so of the root mass fraction, at light intensity of 8 moles per square meter per day. I look at daily light integral rather than the photon flux density, because that's more relevant for growth. And I divide all the observations I have for 8 moles per square meter per day, so all the other levels that were included in the experiment, by the level at 8 moles per square meter per day. So I get a scaled value, which should be 1 at this scaling level of light intensity. And if I do that for all the different experiments, and in total I had 1,650 points which I could use, then I find a line which is basically saturating, right, with an interquartile range, which gives you an idea about the variation that is there among the different experiments. And what I can do then is I can fit a curve through it, and the curve is nicely saturating in this moment. And so I know what the curve is, but that's still pretty complicated if I compare 12 different environmental factors. So what I will show to you is that, I say, okay, I calculate plasticity index, which is the root mass fraction at the scale root mass fraction at a very low light level of 1 mole per square meter per day, and the very high level of 50 moles per square meter per day. So this is where most plants will be growing, somewhere in between. So that's one thing. So in this case, the plasticity index is 1.59. So there's a 60% higher root mass fraction at high light than at low light. But also look, for all these experiments, is the treatment where the plants got the highest light level, is the root mass fraction higher than in the lowest light level. And you see here in 87% of the cases, the root mass fraction is higher for the high light grown plants than for the low light grown plants. So that is a very consistent response. And the closer this value is to 100, or the closer it is to 0, the higher the consistency of the response is. And it also gives you the reliability index, which is a number between 0 and 10, which tells you if we add new information to the database, would I expect that the results would change a lot, yes or no? So this depends on how many data there are in the database, how many different species there are in the database, and how much is the variability among the different experiments. Okay, so I did that for light, as I showed you before. That is this curve here. So the blue line is for the root mass fraction, the brown one is for the stem mass fraction, and the green one is for the leaf mass fraction. And I'll mainly focus on the blue lines, because I think that's easier for now. And you don't need to look at all of them, but basically this is for the rectified ratio, this is UVB, this is the CO2 concentration, and you have ozone, nutrients, water logging and submergence, and temperature and salinity and soil compaction. So basically I only forgot the relative humidity, but unfortunately there are not so many data on experiments with relative humidity, so there was no use to show that to you. Now, what do we see? If you look at these three encircled graphs, these are basically the three conditions or the three environmental factors where the root mass fraction is changing the most. So here you see for light, it is changing quite a lot. For nutrients, it's changing even more. That's the most dramatic response. And for temperature, it is also changing. And then we have a few cases, in the case of water availability and in waterlogging, where we see a kind of intermediate response, and the rest is either responding smaller or it's not responding at all. Okay, so nutrients, light and temperature, those are the three treatments that change root mass fraction the most. And we can list all these things in a table. So when there's a minus sign on the plasticity index, it means that with increasing nutrients, the root mass fraction goes down, right? There's more allocation to leaves and stems. So nutrients and temperature are changing strongly. Irradiance is just changing strongly. And the consistency index is high for nutrients and for irradiance, but it's not so high for the other ones. That means that there are still contrasting responses in the literature, and it may be either because different species respond differently or something else is happening. Okay, now is that a strong response, you may ask, or not? So for light, I looked at 90 different ecophysiological traits, and you see the plasticity index here for all these 90 different traits. And this dotted line gives you a unity, so when there is basically hardly any change, and it's either -1 if the change is negative, or it is positive when the change is positive. And you see that root mass fraction and stem mass and leaf mass fraction, yes, they change, but they are very close to 1, and there are many different traits which respond in a much stronger way over this 1 to 50 moles per square meter per day trajectory, right? So from that perspective, I think that the allocation is only a minor response. And if you look at CO2, for example, we did exactly the same there. And also there you see that the changes in allocation are marginal compared to the changes in a range of other traits. But of course, that's still not quantitative. It's still not really what you would like to know. So we could do also another approach where we say, okay, we know what from this equation in green above, we know that the relative growth rate of plants is basically determined by the physiological components, which is photosynthesis and respiration, and a morphological component, which has to do with how the leaf area leaf weight ratio is, and it has to do with the allocation of biomass to leaves. And when you do the analysis, basically for lights and for CO2 and for water, you see that the unit leaf rate is determining often quite a lot. The specific leaf area can determine quite a lot in the sense that they deviate strongly from this dotted line. But the allocation of biomass to leaf, the leaf mass fraction, is generally very close to the line, which means that it's actually quantitatively not very important. And only when you look at the response for nutrients, now you see that the biomass allocation is really the most relevant response in determining the relative growth rate of the plants. Okay, so my conclusion for the first part is that the changes in allocation, or specifically the changes in leaf mass fraction, are generally smaller than the changes in photosynthesis and respiration and in leaf and root morphology. Okay, then let's go to the second part. So we have different phenotypes of plants, but we also have different phenotypes in phenotypers. And what I told you before is generally what type 1 phenotypers are doing. They are generally simple, happy people who say, okay, we take the biomass of leaves, stems, and roots, and we do this division by the total mass, and then we know exactly what plants invested in leaves, stems, and roots. And here's an example of an experiment we did with low and high nutrients, and where we followed the root mass fraction over time. So we harvested the plants, we chopped them up, we measured the root mass fraction, and at high nutrients, you see the root mass fraction is about 0.32. But when you give less nutrients to the plants, over time, the root mass fraction increases to about 0.5. Right? And that is, in my view, a perfect analysis. But there are other types of phenotypers, that's the type 2 phenotypers, and they are a little bit more critical or gloomy, or how you would express that. And they say, yeah, hey, wait a minute. There is a problem here, because if you apply different levels of an environmental factor, it will mean that the plants will grow differently. And we also know that allocation changes with size. So if you do not control for size, we do not know anything. We have very suspicious data, and the ratios are suspicious anyway, you shouldn't use that. So according to these type 2 phenotypers, there is no insights into allocation if you do not use allometry. And what is allometry? Basically, allometry says, well, we know since a long time that there is a power law that determines the relationship between the mass of organ X and the mass of organ Y. And if we log transform that relationship, we get a straight line. And the interesting thing is that the slope of that line gives the relative growth rates of the mass of the one organ relative to the mass of the other organ. So that's very interesting biological information. And here you see such an allometric plot. So I plotted here the leaf mass plus stem mass on the x-axis and the root mass on the y-axis. And here you see exactly the same data as in the graph before, with the low-end plants in red. And you see that there is, for every shoot mass, there is a difference between the plants growing at high nitrogen and at low nitrogen, right? And basically, this approach of the type 2 phenotypers will give in clear situations the same results as when you do the type 1 phenotyping. But there are, of course, when things are less clear, then it becomes a little bit tricky. And these type 2 phenotypers, they, for example, last 2 weeks ago, there was a paper in New Phytologist where they said allometry is extremely useful for breeding. We cannot do without allometry if you are a breeder. And I'm not completely sure about that. So I would like to plant a little seed of doubt in your mind about the type 2 phenotypers. And there are various reasons I have for that. First, the question is the fit adequately following the data? How about the statistical sensitivity if you compare the slopes of these two lines? To what extent is extrapolation of the data? There is no definition of the size window. And in the end, what does the slope B tell us if we don't know anything about A? So actually, you should consider both the A and the B. And if you want to know whether there really is a difference in allocation, and you find a difference in A, but you also find a difference in B, it could be that the allocation is exactly the same, right? So I think life could be much easier if you go kind of in the middle. So here are the data from the type 1 and the type 2 phenotypers, but we could meet somewhere in the middle and say, okay, we could also plot the root mass fraction as a function of the total mass of the plants. And then you see here the black dots, and you can see here the red dots for the two different treatments. And now if you define a certain window in which you say, okay, in this window from 80 to 160 milligram, for example, I compare the points of the plants of the two treatments, and I can see whether they're significantly different or not. Then you have corrected for size. You know exactly over which time window you're doing that, and you have a much better statistical power than when you try to compare two different slopes. So I think this is much easier, but it is of course still quite interesting to see how does actually the whole allocation change with size, because that is the claim of the type 2 phenotypes. So I teamed up with a number of people whose job it is to go into the forests and to cut trees and to dig out the roots and see how much leaf, stem and root there are in trees in Poland, Spain and in Africa, in Kenya. And I teamed up with a number of people from the Soviet Union and from China who made a huge compilation of the literature of these data, and I have compiled a number of data myself. So I ended up with 11,200 data points about the biomass of leaf, stems and roots. And if I plot it in an allometric graph, where I plot either the leaf mass of a plant against the root mass, or the stem mass against the root mass, or here the leaf mass against the stem mass, then the people that are deep into allometry, they say, okay, we have certain models, and the models, they tell us that there should be the value of B, the value of the slope, should be 0.75. Okay, so I took that to the test. I looked what the slope was. The slope was certainly not 0.75 in this case. For this slope, a value of 1 was expected between the stem and the roots, and that's also not the case. And only in the case of leaf mass versus stem mass, there's a value of 0.74. It's statistically not similar to 0.75, but yeah, okay, you may say, okay, it's very close. So maybe the theory of these of the allometry is still working fine. However, if you look at this line here in the top right quadrant, you see that actually the black line here, most of the points here are below the line, most of the points are also here below the line, and here there are many more points above the line, which indicates that a straight line is probably not the best way to fit these data. And if you're going to check that and you test whether there's also a quadratic part in the equation, you see that for all cases, the quadratic part is highly significant. It doesn't add much to the R square, so it seems insignificant, but if we now fit a quadratic equation and we determine the slope at different parts of the curve, you see that the slope is basically 1 for very young plants, and it goes down to 0.5 for very old plants, right? And this is a range from a few milligrams of plants up to more than 1,000 kilograms of plants, so it's a wide range. But over that range, the theory expects that the value would be constant 0.75, but I think there is not really a ground for that, and actually this slope is changing all the time. So I think that that part of the allometric theory is not working, but still, of course, they have a good point that the leaf mass fraction, stem and root mass fraction may change with the size of the plants. And if I combine all these 11,000 data points and I look what is the leaf mass fraction, in this case we see that for the blue ones are data for herbaceous species. The red ones here you cannot see very well, but they are behind the blue ones, and they are here. That's for the woody species. And what you see that both woody and herbaceous species start with a biomass allocation of, on average, 0.5, right? And then it decreases over time. And by the time that plants get really big, they get over 1 kilogram. So that's basically only trees. Then you see that the allocation to the leaves will go down to 2 or 3%. Now, we can do the same for the stem mass fraction and the root mass fraction, and then you see basically there is a strong exchange between the leaf mass fraction and the stem mass fraction, and the root mass fraction change is more or less the same. It's about 0.3 when plants are young, and it goes down to 0.2 when you have really big trees, right? So there is a lot of variation around it because these plants experience all kinds of different environmental variables. But in principle, there is not really a hidden half. I would say there's at best a hidden 1/3 of maybe even a hidden 20% below grounds. Okay, so if we compare plants with a mass of 1 gram, you see the leaf mass fraction is about 0.5, stem mass fraction about 0.2, and root mass fraction 0.3. So this is a kind of overall average. And if you go to very big plants, you see that trees have about 2 to 3, 4% in the leaves, 70 to 80% in the stem, and only 20% in the roots. Okay, so now we can go back to the data I showed you before about the effects of the different environmental factors on the leaf mass fraction. And if the graph I showed you before is this general graph as any valid also when we look at different acclimation patterns, then we could say, okay, the plasticity index for light was minus 1.2 for the leaf mass fraction. We know that the total dry mass of the plant is changing tenfold over the range between 1 and 15 moles per square meter per day. So when we know this curve, we could correct this response for size, and then the response is not minus 1.2, but is minus 1.13. So yes, there is small change in this case, but the change is not very dramatic. And why is the change not very dramatic? That is because the change in size between plants grown at very high and low light is tenfold, but that's nothing compared to the almost 12-folds, or the order of magnitude of 12 that I showed you for the previous graphs. And we can do that for all the other environmental factors, and basically nothing is changing in most cases, except in the case of water and temperature. You see that the plasticity index may be a little bit higher than what we think just by not correcting for size. And in the case of nutrients, we thought already that the response was quite strong, but the response is even stronger if you correct for the size of the plants. Okay, now we could do a bit more fun things with these data, because we could also think, are there really differences which are among species? So we don't look at the acclimation term as I did before, but we now look at the adaptation. Is there a difference in allocation between different species? And I basically said, okay, what is for each of these 50 groups which I make, where I order all the observations relative to the total mass of the plants, I look what are the species that are above the line and what are the species that are below the line. So within group I make percentiles, and I look what is the percentile values if I analyze that over the whole 11,000 observations. Are there species that are always above this line, and are there other species that are always below this line? Because that could indicate that they're always different, and that is so that there is a phylogenetic difference. So here you see a phylogenetic tree. I don't ask you to read all the names of these plants, but just for your indication, here you see the Solanaceae, so the tomato and other plants, and they generally have a very high allocation to leaves, so a high leaf mass fraction, and at the same time an amazingly low root mass fraction, right? So tomatoes mostly have 8% or 10% of the biomass in roots, and they have much more in the above ground organs. And that's strangely enough also true for eucalypts. In Australia, they also have incredibly high leaf mass fraction. But if you're going to look, for example, at the oaks and the fagus, they have a very high allocation to roots. And that's also true when you go look at the Poaceae, where the grasses and the Cyperaceae, they generally have a high allocation to roots. So in that sense, maybe this artist was a little bit wrong, because he didn't draw that many roots. Okay, now my time is almost up. The organizers would have loved me to talk about sugar allocation as well, but I do realize that only the happy few are able to do these kind of measurements like my colleagues and Jülich do, so I skip it for the moment. And there's one thing I would like to talk with you in the minutes that I have left, and that is about the type 3 phenotypers. And the type 3 phenotypers, that's basically almost all people in this room. And I would like to try to challenge your perspectives a little bit. And last year we already discussed a number of potentials, but also pitfalls when you look at the high-throughput phenotyping platforms. There's one thing that I forgot to mention, or what I didn't want to mention, and I will try to do that now today. And it is, what are we measuring with these high-throughput platforms, and how relevant is what we measure? So when I have these analysis where I try to look at these 100 different traits for species, I try to place them in perspective. So I say, okay, we have a number of traits, you don't need to read it, but they are related to leaf anatomy. We have a number of traits related to the water economy of the plants, to the photosynthesis of the plants. We have traits related to the chemical composition. We have growth-related traits like leaf mass fraction, stem mass fraction, root mass fraction. And then we have, in the end, total dry mass and seed mass and productivity. And how do these traits relate to each other? What are positive and what are negative correlations between these traits? So what is the trait correlation network? And how does every trait affected by growing the plants at higher and lower lights? And by combining these things, you can get a deeper understanding of how the plant is responding to a high light level or a low light level. So all these traits that I compile are about whole plant biomass, biomass allocation, specific leaf area, leaf mass fraction, carbon concentration, starch and sugar, etc., etc. And you can also expand that to the transcriptome and the metabolome and the proteome. They're all kind of hard data. And when I'm here, and I'm talking to many of you, you basically hardly measure any of these traits. So what many people do in the platforms is that in the high throughput platforms, is that they measure digital biomass, or they measure allocation in terms of leaf stem pixels, or they have something like the electron transport rates, or even the transpiration per plant, or they have hyperspectral responses where they have proxies for all these variables. And in fact, there's only 2 traits that I've seen coming by, which are plant height and stem diameter, which are sometimes measured in the throughput platforms, and which are also, which I would say are kind of hard traits, right? So IPPS could also be the plant proxy symposium, because you use all kinds of proxies, and I see the many examples where that is very useful. But my wish, before I leave the floor, is would it not also be possible, if we think about automation, to also think about automation that helps you in destructive measurements? Because is it really sufficient from a scientific perspective that you only watch the plants growing, or should you also chop them up? So I would say if you have machines that can sample automatically the leaves and put them in the -80, or if you have machines that can grind samples at -80, or for example do the root washing, how many hours per year are basically wasted in root washing? And if I dream, I would say even machines that can chop up the plants and determine the leaf area and do the weights. There's a lot of boring stuff that can be automated, and that gives you a lot of hard information. Now, I see when I talk to several of you that some of these machines are working or have been working, and they turned out to be complicated, and I realize it's complicated. But I think that if, for example, you could come up with a root washer which is decently doing its job, it would be wonderful not only to many of us here in the room, but I think to many scientists across the world. So if you, with all your brains, could also find out machines that do destructive phenotyping, I think it would be very cool. Okay, that was basically it. My conclusions are biomass allocation is mainly affected by nutrients, temperature, and light, and not so much by any of the other factors. But the changes, except for nutrients, are generally small relative to the unit leaf rate changes and specific leaf area changes. The type 2 phenotypes are correct that size has an effect on allocation, and basically this is a shift from leaves to stems, and not so much the roots which play a role. Phylogeny plays a role, and I think descriptive analyses are still very relevant for our understanding of the plants. Thank you so much.
Again, we're going to take just one quick question, and we'll have plenty of time for discussion at the end of the session. So you have a question to can read straight about that? Yeah.
Thank you. It's really very nice presentation. So congratulations on that. I don't know if I am allowed to have one very specific question, and very specific also. When it comes to allocation for the biomass. First one, I'm wondering how cactus is, because the leaf is very small and the stem, and then how you consider. I don't see the group of crops there. It's very specific case study. My question more is about when you have parasitic weeds like striga or obenki, and these kind of weeds, they dig nutrients and then they develop depending on the nutrients from the host.
Right.
And when they parasitize the host,
yeah,
they affect the biomass production even in the stem, leaves, or roots.
Yeah.
Is there any, what would be your, thought about that because I did some work and we realized that the parasite affects the biomass production. Why? The question was, or the answer was, to keep water relative content stable. So it affects the biomass, but not the, physiological functioning of the host plant.
I think that's a very cool question. And so what the cacti concerned, cacti are a typical example which produce very few roots, right? So they are in a very dry environment, but they have hardly any roots. And only when the rain comes out, they produce a number of roots, catch the water, and then they shed the roots again. So they have, surprisingly, a very low root mass fraction. And the question you ask about these half parasites, I think that's very cool, but you probably know more about it than I. I have never given it a thought, but it's a very interesting topic. Yeah, thank you so much. Well, I have been challenging you a little bit with the idea that we also should push for destructive harvesting and automation of that. And I would be interested to hear if people see any chance for that, or that it's just too complicated to do that.
Mm-hmm. Yes, that was a very interesting talk. So, the automation triggered me, of course. This plant sampling robot, not the one on the slide, is a product that was developed by a company, ISO Group in the Netherlands. And they sell this robot to breeding companies to take leaf samples. But to me, we've been exploring to integrate this in our own work. But I think the main problem is, but I would like to hear your comment on it, is that if you have an enormous amount of lines developed on tomato, you can make a machine that is suitable for tomato and just take samples every day, thousands per day. But in our case, we got so many different crops and so many different genotypes and species that it's extremely complicated to develop a machine that fits such a chaotic collection of plants. And I think there it goes wrong. There is no return of investment compared to the students, PhD students that do the sampling manually.
Right, so does it apply to the fruits, or does it even apply to punch the leaves of
both? Both. Yeah. So to a robotic implementation of all the manual work that we do in the academic domain, I think.
Yeah, I can imagine that is complicated, but if you do it for one species, that would be helpful for breeders and large companies, right?
Yeah.
Yeah.
So those machines are there. So you can buy those machines. They cost maybe 100,000, 200,000 euros. I'm not a sales guy, so I don't know. But to turn it into an academic useful version,
I can imagine that's a challenge.
Yeah. So yeah, but it's more a comment actually. So
yeah, thanks.
Yeah.
Mm-hmm.
Any other comments or questions related to that? It's good.
Hello, thank you. I loved your presentation, by the way. I noticed that the analysis that you were doing was between woody plants and herbaceous plants, right? But you fitted both of them together, and from the analysis, it looks like there's a clear separation between both. So I'm wondering if you have looked at each of those groups separately and doing the allometry and allocation studies separately for each of those segments of.
Right, good question. So when you look at when you compare the young tree species, the young woody species with the herbaceous species, I found not much of a difference. And of course, this part is only dominated by woody species, so then it doesn't make any sense anymore. But if you compare the woody species and the herbaceous species at a young stage, there's not much difference in the allocation relationship. Right? There are, of course, many differences in other types of traits, but not in this one.
Maybe I'll just jump into this switch. Just to piggyback on the same topic while we're here, I was really interested to see the phylogenetic angle that you had, and trying to tease apart some of this variation by placing the traits onto this large phylogeny. So I noticed that there are still some outliers. So that there are still and so I was wondering, have you looked at other maybe life history traits or other ecological traits that might help to further explain some of this?
Yeah, that's a very good question. So if you look at the Gymnosperms, for example, you see that there are many of the species are color-coded blue. That means that they have a relatively high allocation to the leaf mass fraction. And at the same time, they have, you know, that they have a very low specific leaf area. So those plants have a combination of a very high allocation, very high leaf mass fraction with a low LMA. And that gives a specific leaf area index if you assume a certain density of plants. And if you look in the same group to the deciduous Gymnosperms, they have not a low allocation to leaves. And you have at the same time a lower leaf mass per area, or a higher specific leaf area. So that in the end ends up in a quite similar leaf area index at a given density here. So I think that it's not only purely the phylogenetic constraints, but there's also a functional constraint between the evergreens and the non-evergreens. Yeah, so there's more in that direction than.
Yeah. I guess just similarly, still in this, so trees and shrubs, they are often easy to tell apart, but sometimes a given species, you can find it with either growth form. Have you looked at species that can grow either way?
Nobody measures shrubs, almost nobody. So there are trees in there. There are herbaceous species in here, but really the shrubs are an extreme minority. So it's difficult to say anything about that group. I expect they more or less go with the woody species, but with all the woody species. But there are not enough data to check that.
Okay.
So continuing that conversation, I'm going to ask you to speculate just a little bit. When you start thinking about allometry within the plants, you can start thinking about functional versus structural components within it. So root tips versus the rest of the roots. How would that change within the structures, and what would you expect with the allometry changes?
So you would like to separate fine roots from coarse roots?
Or expand from functional leaves?
I would expect that for function for fine roots compared to coarse roots, if you have more fine roots, you basically must have more coarse roots as well. And to function the fine roots in separate places in the soil, you also need to have your coarse roots to kind of position them in that place. So I presume that there is a kind of allometric relationship between those two, that you have to have more coarse roots if you want to make more fine roots. We had a workshop about it last week, actually, and it turns out that also there it's very difficult to find the data for plants. And if you have, there are some data, but then they are for pretty young plants, and you would actually like to know that for all the plants, right? But there's a very shortage of data in that respect. So I can't answer your question. But you would expect that there is an allometric relationship between the two.
Yeah, thank you very much, Hendrik, for your nice presentation before. I like a lot the slide where you challenge phenotypers, saying that there is a kind of gap to bridge between, yes, exactly, between morphological traits, that is basically what we estimate with high-throughput plant phenotyping, and the more functional traits that would be necessary to characterize crop functioning at the species level, at the genotype level. So what do you think that we need to bridge this gap? Is only up to the phenotypers to do all the job to come through the functional traits? Are ecophysiologists who need to understand or how to benefit from the high throughput phenotyping, or there is an element probably by another scientific discipline that we have to put in the middle to bridge that gap? What is your opinion about it?
Yeah, good question. So first I want to stress, I hope that I told before that I think that a lot of these non-destructive measurements are very useful in various different settings, right? So it's only when you push from a scientific way, you try to understand these things, that I think that these high traits are really bringing us forward. Now, as far as where that has to come from, I guess that the phenotypes are generally richer than the ecophysiologists. So for them, it would probably a little bit more easy. Yeah, but I think for the ecophysiologists, they're more motivated to do these kind of things, right? So I still hope that these people can team together. And what you see that all these phenotyping systems started actually in agronomy, but they are now going into the ecology as well. So I hope when it reaches the more ecological-minded people, that the ecophysiologists there also have their push to make these things happening in the direction of the high traits. But I realize it's a difficult thing, but I tried at least to make you aware of that there is a wealth of interesting traits that we would like to measure as well. But it's not easy. I mean, even if you measure the transpiration rate per plant, right, that is already a very nice ecophysiological parameter. But still, the plant can be small or it can be large. And yeah, we are used to kind of scale that by dividing it per unit leaf area. And I think now it is possible if we scan the plant itself, and we know the water loss of the plants, to calculate what the transpiration rate per unit leaf area is. So I think that is what I see from here now. That is quite in reach, right? So that's something we can easily achieve. How it works when we go to these hyperspectral images and see to what extent we can determine starch and sugar, etc. That's, I think there's still a stronger gap between having the really right measurements and predict that very well. And the same is, I think, spot meters can determine the chlorophyll content very nicely. But for example, they don't look at the chlorophyll A to B ratio. And I have no idea whether that would be feasible anyway. But if you would know this A to B ratio, you could characterize what the light environment is that the plants experience. Or you have, to what light environment just. And so I think there is information where we are close, but we are not making the last step yet. And I hope that the bright minds here are able to do that in the future. Is that a little bit, it's not a fully satisfying answer, but...
Yeah, just to continue on your answer, there are some systems that predict some of your ecophysiological traits with a certain accuracy, but then nobody finds what is good enough. So if you can estimate the whole plant biomass, or you can estimate the sugar content in a leaf with a R-square of 0.6, can you use it? Or is it only good enough if you go beyond 0.8? Can you provide some guideline that you say, okay, this is useful, this is not good enough?
So, when you look at our paper that we wrote, R-square is a very difficult thing to use because it depends on the variation you have in the X and the Y axis, right? So the longer you calibrate with the wider the variation, the higher your R-square is. But maybe you just want to apply it at the final harvest to see what is the biggest plant. And then your R-square is not 0.99 anymore, but your R-square is much lower. So maybe we should get away from simply looking at the R-square and look at what percent difference there is between the real observations and the line that you use for calibration, and focus on the percentage mistake you make or the percentage accuracy you have in that way, rather than on your R-square. And then I would say it would be cool if you have an estimate which is within 10% of the real value. I think that is generally good enough to work with. But if the difference is 20 or 30%, then it's still not so good. Is that okay?
And Henrik, I'm wondering if to that intersection you could also add things like relative growth rate or absolute growth rates, which, because the power of a lot of the facilities that we have now is that time course series measurements that we have and that non-destructive measurement, and I'm sort of missing that relative growth rate in that intersection, because yes, we may measure pixels with one system and biomass with the other, but the relative growth rate is, often a common one.
Yeah, I think good points. I think the non-destructive systems are actually very powerful to measure relative growth rate, because that is especially a trait where if you do destructive harvest over time, you get a lot of variation, and it makes that estimate not so good as when you do that in a non-destructive system, until of course you get overlapping of leaves. And then if you have a rosette plant like Arabidopsis and there is so much overlap that you don't see the new leaves anymore, then your RGR is underestimated by the system you use. But before that time, I think RGR, yeah, you're right. That should be one that should be in.